Hello and welcome to my channel Omni Bioentrance, and today we will be discussing the CSIR NET JRF Life Science question paper. This is part C of July 2025 question paper. So coming to the first question, if a 10 microgram of pure carbonic anhydrase catalyzes the hydration of 0.3 grams of carbon dioxide in 1 minute at 37 degrees Celsius at Vmax, what is the turnover number of carbonic anhydrase? Okay, so here you have to calculate the turnover number of carbonic anhydrase. They have given molecular weight of carbonic anhydrase, how much microgram is present, and it catalyzes the hydration of 0.3 gram of carbon dioxide in 1 minute at 37 degrees Celsius. So mass of carbon dioxide, time, mass of enzyme, molecular weight of carbonic anhydrase is given. So Kcat is given by the formula moles of carbon dioxide produced by moles of enzyme into time. So first we have to calculate moles of carbon dioxide. So they have given, we know the molar mass of carbon dioxide, that is 44 gram per mole. So moles is equal to weight by molecular mass. So that will give you 6.82 into 10 raised to 3 moles. Now calculate the moles of enzyme. They have given the mass mass of enzyme as 10 microgram. So convert it into gram. So 10 into 10 raised to minus 6 gram by molecular weight also they have given, that is 30,000 grams per mole. So you will get a value. Now substitute in the Kcat and you will get 2.05 into 10 raised to 7 per minute. So that will be 2 into 10 raised to 7 to 2.1 into 10 raised to 7 per minute. Then next one. A canonical Watson-Crick GC base pair is stabilized by 3 hydrogen bonds formed between a donor and an acceptor atom located at a distance. So this is the donor and this is the acceptor. This is the distance between the two. Assume that the dielectric constant D in the DNA core is 4 and in the water it is 78.5. Which one of the following options correctly represent the total difference in electrostatic interaction energy for all 3 hydrogen bonds when buried in the DNA core versus fully exposed solvent? So you have to find the difference in electrostatic interaction when this canonical Watson-Crick is base pair is buried in the DNA core as well as when it is exposed to the solvent. For all the 3 hydrogen bonds you have to find out. So what is the difference in electrostatic energy? So charge on the donor, it is already given, and the acceptor also they have given. The distance between these 2 charges is also given. The dielectric constant in the DNA core and in water also given. K is the constant, that is also given. Now you have to find the difference in electrostatic energy. For all the 3 hydrogen bonds you have to find. So electrostatic interaction energy, it is given by the formula K into Q1 into Q2 by D into R. So for the DNA core, for 1 hydrogen bond, substitute the values for dielectric constant, use 4 and you will get minus 104.7 kilojoule per mole. Then for electrostatic interaction energy in water for 1 hydrogen bond. So in water means the dielectric constant is 78.5. So substitute that and you will get 5.34 kilojoule per mole. Then difference in energy, you have to multiply it by 3. This difference you have to multiply by 3 and you will get minus 298.1 kilojoule per mole. Then next one, gastric juice, that is pH 1.5 is produced by pumping HCl from blood plasma into the stomach. calculate the amount of energy required for H plus transport to produce 1 liter gastric juice at 37 degrees Celsius. So you have to calculate the free energy required to transport H plus from blood plasma into the stomach to create a pH of 1.5 in the gastric juice. The blood plasma is at pH 7.4. Then that also you have to calculate. You have to calculate how many moles of ATP must be hydrolyzed to provide this amount of energy. So the free energy for ATP hydrolysis is minus 58 kilojoule per mole, and joules are the constant, gas constant is 8.315 joule per mole per Kelvin. So first pH of gastric juice they have given, and pH of blood plasma also given. T they have told it is 37 degrees Celsius, so convert to Kelvin because R is in Kelvin, joule per mole per Kelvin. So 310 Kelvin. So find the free energy change. You have the equation 2.303 RT into delta pH. So substitute the values and you will get approximately 35 kilojoule per mole. Now you have to calculate how much ATP is hydrolyzed for this. So delta G of ATP hydrolysis, it is already given, that is minus 58 kilojoule per mole. So moles of ATP required will be total energy by delta G ATP hydrolysis. So that will be 35 divided by 58. So that will be around 0.6. Okay. Then next one, the primary sequence of miRNA is given here. The seed region of miRNA starts from second position, that is here, to the 8th. 1, 2, 3, 4, 5, 6, 7, 8. This is the 8th position. The seed region is the one where this miRNA will form complementary base pairing with the mRNA. And after forming this complementary base pairing with mRNA, it will degrade the mRNA with the help of certain enzymes. So which of the following mRNA sequence would be targeted by the above miRNA? So the miRNA is going to target a particular mRNA sequence. So this mRNA sequence should have a region which is complementary to the seed region of miRNA, so that it can form complementary base pairing with the mRNA and bring this mRNA for degradation. So here 5 prime AG, I have written it here, AG, C, UU, AU, CA. I have not written C and A, but you will get the answer. Okay, let me see, 1, 2, 3, 4, 5, 6, 7, 8. Okay, C, till C it is not A. A is not included till C. Okay. So here one C is there. I have not written that C, and it forms base pairing with G. And this A is not included, only till C. That is the seed region. So A, G, C, UU, AU, 3 prime, 5 prime, which is U, C, G, AA, UAG. So if you see the first sequence, you can see somewhere here, that is GAU, AA, GCU. That is the region complementary for this particular miRNA. In no other mRNA you will find this sequence. Okay. So this is in the 5 prime to 3 prime direction. So you have to search from this way. Okay, in your mRNA, in this complementary sequence. So GAU, AAG, CU. Then next one, given below are the thermal melting curves for proteins A, P, Q and R. This x-axis it denotes the temperature, and the y-axis it denotes the concentration of unfolded proteins. Now you have to see the state which is true regarding thermostability of the protein A with respect to P, Q and R. So this thermal melting curve, what it tells us is the denaturation process of a protein as the temperature increases, or unfolding of the protein as the temperature increases. And the Tm, that is the temperature at which half of the protein is unfolded. That is, it has lost its native state and activity, but Tm. So if the Tm is higher, okay, remember this, if the Tm is higher, that is, if it requires a higher temperature to unfold 50%, then it is more thermostable. It requires higher temperature to undergo unfolding to 50% condition, means it is more thermostable. So a higher temperature means greater thermostability, or I can say the protein requires more energy to denature. So it is more thermostable. Now come to the problem. So if you see this bold line is A and this is your P, Q and R. Here it is P. The dotted line is P here and this line is A. So if you see P and A, you can see that this Tm is almost the same, that is 50%, or unfolding is at a similar temperature. Okay, at around the similar temperature, they have the same temperature or same Tm value for unfolding. That means they are almost similar in thermostability. But when you compare A with Q, you can see that only when the temperature has increased. See, A requires lesser temperature, Q requires more temperature to unfold 50%. Then it is Q is more thermostable than A. Similarly, R also, it requires more temperature to unfold than A. So R is more thermostable than A. Now see the options. So P and A, it will be almost same. That means 1 gone, 2 gone, 1 also gone, 2 also gone. So 3 and 4. Okay, P is A approximately to P. Okay, A is less than Q, A is less than R. Yes, that is correct. 4 they have given A greater than R, which is not correct. So the answer is option 3. An alpha helix formed by 30 residues is stabilized by 25 backbone hydrogen bonds in its folded state. So alpha helix is 30 residues and there are 25 hydrogen bond in the folded state. Assume that average energy of hydrogen bond is minus 10 kilojoule per mole. And the disruption in the peptide resonance causes a partial loss of 205 kilojoule per peptide bond. Assume that the folding and unfolding process only changes peptide resonance stabilization partially and the loss of hydrogen bond upon unfolding. Which one of the following option represent the magnitude of net energetic difference between the folded and unfolded state of the helix? So the average energy of hydrogen bond is 10 kilojoule per mole. So to break one hydrogen bond, you need this much energy. And peptide resonance disruption, it will result in 205 kilojoule per mole per peptide bond. Okay, for unfolding, upon unfolding. Okay, now you have to calculate the net energetic difference between folded and unfolded state of the helix. So first, the total energy contributed by hydrogen bond in the folded state. So number of hydrogen bond there are 25 and energy per hydrogen bond they have given in the question, it is 10. So energy contributed by hydrogen bond is 250. That means that much energy has to be supplied for the protein to be unfolded. Okay. Now peptide resonance stabilization energy, that is disruption of peptide resonance. It will cause a release of 205 kilojoule per peptide bond. Now here the length of the peptide chain is 30 residues. So number of peptide bond is 30 minus 1, that is 29. So energy lost by peptide resonance is number of peptide bonds into energy per peptide bond. So 29 into 205 kilojoule per mole, you will get this one. Now energy difference is energy contributed by hydrogen bond plus energy lost by peptide resonance, that is 6195 kilojoule per mole. Now these values, that is energy contributed by hydrogen bond and those lost by peptide bond, are added because they both are stabilizing energies. Okay, that is they stabilize the folded state of the protein and they are lost. These energies are lost upon unfolding. So you calculate the net difference between the folded and unfolded state by adding up these energies. Okay. Now come to the next question. Spindle assembly requires the formation of numerous new microtubules. Okay. To meet this demand, spindle contains large number of regulatory factors. A few protein or protein complexes are mentioned below. So microtubule nucleation, which of the following options are all the factors that directly impact microtubule nucleation for spindle assembly? Microtubule nucleation means the initial formation of these microtubule polymers, and the polymers which are involved in microtubule formation are your alpha and beta tubulins. Okay. So first one is gamma TURCS. So it is gamma tubulin ring complex, and it serves as a template for the assembly of the first alpha tubulin and beta tubulin, and thereby it will initiate the growth of microtubule, because microtubule nucleation means initial formation of microtubule polymers. Then so that is involved. Then there is augmine. Augmine, it is found on the surface of already existing microtubules, and what they do is they will recruit this gamma TURCS to this spindle and thereby promote nucleation. Then there is, so that is involved. Then there is TPX2. TPX2 is also promote nucleation and elongation of microtubule at the kinetochores as well as at the spindle poles. So C is also involved. D is dynein. It is not involved. It is a negative end directed motor protein, and it is involved in retrograde transport. It is not involved in nucleation of microtubules. So A, B, C is the answer. Answer is option 3. Then protein possess a strong sorting signal known as nuclear localization signal for their import into nucleus. Okay. So protein which are directed to the nucleus, they will have this NLS on them. So a few statements are given below on the import of protein destined for nucleus. Which are the correct statements? So this is the protein which is destined for transport to nucleus. So this is the nucleoplasm, this is your cytoplasm. Here there is the nuclear pore through which this protein will be exported to the nucleus from the cytoplasm. So this protein which is destined for nucleus will have the NLS signal sequence. Now this NLS signal sequence binds the alpha importin. So first to bind will be alpha importin. Alpha importin will direct the binding of the beta importin in the cytoplasm. So this whole complex, it will move through the nuclear pore to the nucleus. Now the nuclear pores have this protein called as nucleoporin, which is having phenylalanine glycine repeats. This protein will recognize this importin beta and it will facilitate the movement of this complex bound to the protein destined to nucleus through the nuclear pore. And these interactions between this importin and the nucleoporin subunit is of low affinity. Okay, and through that nuclear pore, it will reach the nucleoplasm. And once the protein reaches the nucleoplasm, RAN GTP, it is found in more higher concentration in the nucleoplasm than in the cytoplasm. So RAN GTP, it will cause the dissociation of this protein from the alpha and beta importin, and the protein will be released in the nucleus. Okay, now see the statements. The importin receptor binds to the nuclear localization signal. Yes, importin, the import receptor is nothing but your alpha and beta importin. They will bind to the nuclear localization signal, that is correct. Then the import receptor contains several low affinity binding site for the nucleoporins. Yes, I told you nucleoporin is having phenylalanine glycine residue to which this importin proteins can bind, and it is by the low affinity that they are binding to this nucleoporin. So that is also correct. The nuclear import receptor cargo complex, that is the nuclear protein which is destined to the nucleus, as well as which is and import receptor means bound to alpha and beta importin. So that complex, that is the protein with alpha beta importin complex, is dissociated by RAN GDP. No, it is RAN GTP that is present in the nucleus, not RAN GDP. So C is wrong. The nuclear import receptor cargo complex binds to RAN GTP in the cytosol. No, it doesn't bind to RAN GTP in the cytosol. Actually, the import receptor cargo complex is formed in the cytosol, and RAN GTP binds when the complex enters the nucleus. So D is also not correct. Then next one. The following statements are made regarding vesicle formation. Okay, you should remember the snare protein, V snare, T snare, then fusion of the vesicle with the target membrane. All these things you have to remember. So vesicle, this is with vesicular transport. Transport and fusion in cell expressing a mutant ARF protein. So the protein ARF is mutated. This mutant ARF protein is constitutively bound to GTP. So the mutation for the ARF protein is such that it will always be bound to GTP. It is defective for GTP hydrolysis. It doesn't hydrolyze its GTP back to GDP. So they have asked which are the correct statements. So first we will see the mechanism of COP1 transport. So first COP1 vesicle formation. First the vesicle has to be formed. This vesicle formation is before the fusion with the target membrane. Okay, so these steps are taking before fusion with the target membrane. So COP1 vesicle formation, it starts when ARF1, okay, ARF1 bound to GDP. This is the form. I have drawn two round here. That is ARF1 bound to GDP attaches to the GBF1, another protein, and upon attaching there, GBF1 is a GTP exchange factor. Okay, yes, guanine nucleotide exchange factor, and it catalyzes the activation of ARF1. How? It will promote the exchange of this ARF GDP for GTP, and once ARF1 gets bound to GTP, there will be conformational change in the ARF. See here, first it was circle, now it has become rectangle. So as a result of this conformational change, the ARF1, it will expose a myristoyl group. Now with the help of this myristoyl group, this is the plasma membrane, it will bind to the membrane. Okay. So ARF1 GTP will bind to the membrane. Now this bound, once this is bound to the membrane, it will recruit the ARF GAP1 COP1 proteins. Okay. And this whole, that is ARF1 GTP, ARF GAP1 and COP1, they all will bind to the cargo protein, forming a complex. And this will induce the membrane to form a vesicle. This is the plasma membrane forming a vesicle, and all these are bound to the plasma membrane. And finally, the vesicle is completed, means it will bud off from the membrane. Okay. And that portion I have not shown, where the plasma membrane is bound to all these proteins and forming a vesicle. Okay, that portion I have not shown. I have not shown the budding actually. Formation of the vesicle I have shown. Now once this vesicle formation is completed, the ARF1 GTP will promote the release of or hydrolyze the GTP to GDP. Okay, ARF1 will get hydrolyzes its GTP to GDP. With that, what will happen? The complex will be broken. So ARF GAP1 will be released, COP also will be released, ARF1 GDP bond will come back to its original conformation. Now all these things will be released and your target protein will be bound to the vesicle for going and fusing with the target membrane. So this is the first stages of vesicular transport. Now come to the question. So in the question they have told it is a mutant ARF protein and it is defective for GTP hydrolysis. That is, it is constitutively bound to GTP. Now once this ARF protein is constitutively bound to GTP and it is GTP hydrolysis defective, means the vesicle will be formed, but the vesicle will not be able to fuse with the target membrane. Why? Because if the vesicle has to fuse with the target membrane, the COP1 coat has to be removed off or disassembled from the vesicle. So since this ARF protein is bound to GTP continuously and it cannot be hydrolyzed to GDP, it will always remain bound to COP1 coat, and the COP1 coat will not be disassembled from the vesicle, and the vesicle will not be able to go and fuse with the target membrane and deliver the cargo. Okay. Now read the options. COP1 coated vesicles are only formed from their normal location. Only. So, no. It is not like that. It is a mutant. It is constitutively bound to GTP. So, it need not be from their normal location. It can be from anywhere. Okay. It need not only from the normal location. It need not be. Okay. Then ARF1 is constitutively bound to membrane. Yes. First ARF1 GDP, it is not bound to membrane. Once the GDP is hydrolyzed to GTP, it is able to bind to the membrane. That is what we saw in the normal condition. Now mutant ARF protein, it is always bound to GTP, so it will always remain bound to the membrane. It will not be released from the membrane. After the formation of the vesicle also, in normal condition, ARF protein will hydrolyze its GTP to GDP, and it will result in the release of the COP1 coat. But here it is mutant and it is always bound to GTP. It will always remain bound to the membrane. So B is correct. So A is wrong. So 1 gone, 2 gone. Then COP2 coated vesicle fusion with the target membrane is aberrant. Now COP2 coated vesicle fusion, it is not regulated by ARF protein. It is by another protein called as the SAR1, SAR1. So a mutant ARF protein is not going to affect the COP2 coated vesicle fusion. So it is incorrect. Then COP1 coated vesicle fusion with the target membrane is disrupted. Yes, because the COP1 coat is not disassembled from the vesicle. Once it is not disassembled from the vesicle means it will not be able to fuse with the target membrane. So fusion will be disrupted. So answer B and D only. 4 is the right answer. Then next one, the table below describes the effect of various antibiotics on protein translation in bacteria. Okay, erythromycin. Erythromycin, it binds to 23S rRNA in the 50S subunit of the ribosome. 100% correct. A1, you have to know these antibiotic and their mechanism of action by heart. No other option. So A1. Then chloramphenicol, again the one which bind to the 50S subunit and inhibits the peptide bond formation. So chloramphenicol bind to 50S subunit inhibits elongation of the peptide chain. Yes, new peptide bonds will not be formed. So B2. That means option 1 is the right answer. See here B3 they have given, so that is not correct. A3, A2 again wrong. So what is left out? 1. Then tetracycline. Tetracycline I think to the A site. So new amino acylated tRNA will not enter into the A site. So it binds to the 30S ribosomal subunit, interfering with binding of amino acid tRNA. Yes. C3. Then streptomycin, it will inhibit initiation of protein synthesis. So bind to 16S rRNA. Yes, 16S rRNA is required for complementary base pairing with the mRNA, because 16S rRNA is the one which is having your Shine-Dalgarno sequence. So it binds to 16S rRNA means mRNA will not be able to come and bind there. So initiation of protein synthesis is blocked. So in the 30th subunit of the ribosome. So D4. Then next one, eukaryotes regulate gene expression via histone modification. So in the table given below, histone modification column X, its association with the chromatin type, that is in column Y, and whether gene is on or off in column Z. Now you have to match these. So you know histone modifications can be methylation or acetylation, and because of this histone modification, the structure of the chromatin will be altered, and it will in some cases it will result in the DNA getting accessible to the transcriptional machinery, and in other cases the opposite of that will happen, that is the DNA, that is in case of some methylations, the DNA will remain in the heterochromatin state, and it will not be accessible to the transcription machinery, and it will remain, gene expression will be off. Now come to these. Here also you have to by heart all these many of these histone modifications and what they do, whether they turn on the gene or off the gene. In so first one, H4K4Me. So histone 4 at lysine 4th position is methylated. So that means it is highly accessible open chromatin. Highly accessible means maybe your promoter region of actively transcribed genes. Okay. So it is highly accessible open chromatin and gene expression is on. So A, that is first answer itself is the right answer. Here it is A, B, A, C. First one, if you know, the answer is ready. Okay, then H3K9Me. So H3K9Me, it is always associated with both the constitutive and facultative heterochromatin. So this modification will result in heterochromatin. That is, it can be both constitutive or facultative. In majority of the cases it is constitutive, but both constitutive and facultative heterochromatin are seen. And heterochromatin means gene expression off. So BC2, okay. Then H3K9AC, acetylation. Again, it is found in highly accessible open chromatin. So gene expression is on. So CA1. Then H3K27Me, it is the hallmark of facultative heterochromatin. This one, H3K27Me, it is hallmark of facultative. So D, only facultative heterochromatin and it is off. Heterochromatin, so gene expression is off. So answer is option 1. Then next one, they have given some lipids and they have asked for their chemical structure. So those lipids I am showing it here with their chemical structure. So first one, it is a sphingomyelin. It is having a ceramide backbone to which a phosphocholine group is added. Okay, that is with case of sphingomyelin. Then you have the archaeal membrane, that is it has ether linkage. See here, ether linkage and not ester linkage as in the case of bacteria. In archaea it is isoprenoid chain and in bacteria it is fatty acid chain. So that is the difference, that is with case of archaeal membrane. Then you have got the triacyl glyceride, that is to the glycerol backbone 3 fatty acid chains are added or will give rise to triglyceride, and these lack any polar residues, so it is a non-polar one. Then you have got the plasmalogens. So here the glycerol backbone is there. Then this to the first carbon means that position is called as SN1. The second carbon it is SN2 and the third carbon it is SN3. Now if in the first carbon it is the vinyl ether bonding. Okay. It is an ether linkage. Whereas in the second SN, that is the SN1 position. Then this is the SN2 position where you have a PUFA attached via ester bond. PUFA is arachidonic acid. So an ester bond is formed and it is at the SN2 position. Then a phosphate group is attached at the SN3 position. Okay, that is in the case of plasmalogens. Then you have the cerboside, the same ceramide backbone to which a simple sugar molecule, and if it is a cerboside, it is a galactose that is added. Fine. Now come to the match the following. So given below are a few biological lipids in column X and their chemical nature in Y. So you have to match. So sphingomyelin, they have a ceramide backbone with a phosphocholine. So okay, phosphocholine head group. So A4 that makes 1 and 2 gone. 3 and 4 we have to see. Then RKL membrane, they have ether linkage, or I can say they have very long branched chain fatty acid. Okay, yes, they are isoprenoid chains, so they are very long branched chain fatty acids. So B will be 5, that makes option 3 as the answer. Then C3, non-polar lipid triglyceride. We already discussed that. So C3 and plasmalogens D2, that is ether linked alkene. Yes, that is your vinyl ether group. So it is a ether linked alkene. So D2. Okay, D2 plasmalogens, it is ether linked alkene, and cerebroside galactose is attached to the ceramide. So E1. So that makes option 3. So at present, this many questions I have discussed with the part C. I will come up again with the rest. Till then, thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of CSIR being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, C for CSIR, and you will find them there. Thank you.